Hi everyone! Thank you so much for watching this video. My name is Sarah and this is Lydia.
Hi.
We are PhD students in the Health Psychology doctoral program here at UNC Charlotte. And today we're holding a virtual review session to go over the material for Exam 1 in your Psych 2113 Introduction to Brain and Behavior course. This video will be about 45 minutes to an hour long, and we hope to cover some of the most important or, I guess, notoriously difficult parts of the exam in this video. So before we get started on the material, we wanted to just go over the format of the exam. So the exam is approximately 50 to 60 questions long, and it consists of 3 different question formats. It's true, false, multiple choice, and matching questions. So keep in mind that some of the questions require multiple answers. So for example, if you have just one matching question, there could be 5 or 6 concepts that you need to match. So keep that in mind when you're thinking about your pace or your timing for taking this online exam. The exam is going to be open for 72 hours. That means that you'll have an entire 3 days to take this exam or start the exam. But once you start it, you only have an hour to complete it. So the start time of that window for Exam 1 is listed on your syllabus, and Dr. Levins will also have made that very clear in her lectures when that 72-hour window starts. So keeping in mind the 50 to 60 question length and the hour-long time limit that you have once you start the exam, this gives you around 1 minute per question. So most students really do finish on time and don't have a problem completing the exam or doing well. However, some of the students have said that it is difficult to complete this online exam unless you are very well prepared for it. So an online exam doesn't mean that it's an easier exam. In fact, in some regards, students have said that these exams are very rigorous and actually more difficult than typical in-person exams. So you won't have time to be thumbing through your notes or the book while you're taking the exam. So it's really important that you study hard before you sit down to take it. It's also important that you take the exam at a time when you feel alert and you're in a work-like environment. So for example, it'd be best to probably take it in the morning at some point at a desk and not take it at maybe 11 p.m. sitting in your bed. So try to simulate how you would normally be taking an exam, an in-person exam.
So in this review session, we're going to be taking you through the study guide for Exam 1. It's posted on Moodle. It's the PDF, and it's posted in the Midterm 1 and the review materials section. So we're going to go over the kind of condensed version of everything that you've learned in the past several chapters. So we do recommend that you have the study guide open in front of you while you watch this video. We're also going to have it open in front of us to help us kind of facilitate the study review session. So specifically, this exam is going to cover these chapters. Chapter 1, which describes the origin of brain and behavior. Chapter 2, which reviews how the nervous system functions. Chapter 3, which covers the functional units of the nervous system. Chapter 4, which covers how neurons use electrochemical signals to transmit information. Chapter 5, which reviews how neurons use electrochemical signals to communicate and adapt. And then finally, Chapter 8, which describes nervous system development. So we're not going to be going over all of this information in great detail because there's also a detailed study guide and you have access to all of the lectures, but we'll just be covering the most important things and the notoriously difficult aspects of the exam.
So now we're going to get into the actual study guide. So we're going to bring it up and we're going to walk through the study guide and highlight some of the key aspects that you should know and that have been difficult for this exam. So this is our first video that we're recording as part of these virtual review sessions. So just bear with us and we're going to start that review right now.
So now we have the study guide open. I'm just going to kind of scroll through these first few pages from the chapter 1 because we're not going to be emphasizing that really right here. It's not the more technical information, so Just make sure that you know all of this, but we're just not going to be covering that in detail. So we're going to start off talking about basic nervous system anatomy. This is information from Chapter 2. So the nervous system is comprised of both the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord, and the peripheral nervous system consists of the neurons that run outside of the CNS to the muscles, organs, and the skin. It has two main branches down here. So it has the somatic nervous system. The somatic nervous system and the autonomic nervous system, which are the voluntary and involuntary. So then within that autonomic nervous system, we have the sympathetic nervous system and the parasympathetic nervous system. The sympathetic is what drives physiological activation, or that fight or flight responses, while the parasympathetic nervous system inhibits that activation and prepares the body for rest. And then remember, when we're talking about, so I'll go back down here, when we're talking about neurotransmitters in the autonomic nervous system, there's afferent and efferent information. So afferent is incoming information and the efferent is outgoing. So you can remember that by the efferent meaning exiting. So now we're going to go over the divisions of the brain, which is just a continuation of Chapter 2. So you'll definitely need to know the divisions of the brain. So it's going to be the forebrain, the midbrain, and the hindbrain. The study guide goes over every part that you should know, but just basically make sure that you know all the functions of the subdivisions, and then we'll be covering the functions of some of the brain regions in a moment. So for example, you will need to know that the medulla oblongata is in the hindbrain, and it's responsible for vital reflexes like breathing, heart rate, and salivation. You can also remember that this is the medulla oblongata because it is oblong. And then you will also need to know about the 4 lobes of the cerebral cortex. So these are here. So this is the frontal lobe, this is the parietal lobe, the temporal lobe, and the occipital lobe. So the frontal lobe is responsible for the motor and executive functioning. The parietal lobe is responsible for processing tactile information like touch, movement, attention, and spatial functions. And then the temporal lobe is primarily responsible for processing auditory information like sounds and speech. And then the occipital lobe is primarily responsible for processing visual information. And you can remember that by thinking about ocular glasses that help people to see better. And then just remember at the end of Chapter 2, there are also those 10 principles of nervous system function, so make sure that you know those.
All right, now we're switching over to one of the worksheets that you have posted in your Exam 1 and Review Materials section. So this is the Basic Brain Anatomy Study Guide. So hopefully you guys have all filled this out by the time you've watched this video, and so we're kind of just going to be reviewing some of the answers to that. So the first page goes over specific areas of the brain, their locations, and their main primary functions. So as opposed to just sort of reading through all of this, I'll pull out some of the more important or rather confusing ones. Oftentimes people get the thalamus and hypothalamus confused. So the thalamus, they're both located in the diencephalon, which is technically in the forebrain area. And the thalamus is a relay station for sensory organs and input to the cortex, while the hypothalamus really communicates with a number of different things. So I think one of its primary functions is to govern hormones. So it communicates with the pituitary gland, which is the master hormone producer, and it helps to regulate things in our body like temperature, eating, drinking, and sexual behavior. And so sometimes we think about those as the 4 Fs. So you can remember that for the hypothalamus, that it's sort of a master of all of these different types of systems. Another one to focus on is the corpus callosum or callosum. This is a white band that runs along the length of the cerebral hemispheres, and it's important because it helps the 2 halves of the cortex communicate together. So we're not going to go in too much detail about this, but I would highly recommend filling out the study guide on your own and making sure that you know all of the different areas of the brain, but also how they work. Sometimes it's helpful to use this information in conjunction with the next page of this worksheet or study guide, which details the major areas of the brain. So if you can think about the major areas, what they do, and also how they interact, then you'll likely know all of the information that you need to do well on this portion of the exam from Chapter 2 about neuroanatomy. All right, so switching back to the study guide, Chapters 3 through 5 cover a lot of important material for this exam. So we're going to be going over first information from Chapter 3, which include the anatomy of a neuron, types of neurons, and the cells that support neurons. So starting with the anatomy of a neuron, this is a great diagram on the study guide right here. I also wanted to point out this link that's at the bottom of the diagram. This is a link to a website that's a great resource for reviewing things about neurons, how neurons communicate using electricity and chemicals, and good information about neurotransmitters. So I'd recommend going to check it out. There's a video on there, and there are different exercises that you can do. Plus, the website has more chapters that could help you review information that you might see on exam 2 or 3. So, all right, so looking at this diagram, we can sort of think about the start of the anatomy of a neuron with its dendrites. So there are a number of dendrites, and these structures gather information from other neurons. Then the cell body, also known as the soma, integrates this information and sends it down the axon. So the axon is a passageway that sends information from the cell body to other neurons, muscles, or glands. And remember, a neuron can have many dendrites, but it only has one axon. An axon can also have a myelin sheath, or be what's called be myelinated, and that covers the axon of some neurons and helps speed up neural impulses. So the more myelination an axon has, the faster the information will travel, or the impulse will travel. And then finally we have the terminal button, which forms junctions with other cells. And so this diagram is great because it sort of shows how two neurons might communicate with each other, because these terminal buttons link up with other dendrites from another neuron, and it shows how we can start to see how communication happens between neurons. So to recap, information from other neurons is collected at dendrites, it's processed in the cell body, and then passed on to the axon, and then finally it's passed to the terminal, where it is passed on to a target. So that was a broad overview of neurotransmission, and I want to point out that we do have action potential listed here, but we're going to talk about that in more depth when we go over Chapter 4 information. Other Chapter 3 important info to know are these 3 types of neurons. So there are 3 main types. Sensory neurons, these are the simplest types of neurons. They bring information into the central nervous system. Then we have interneurons. These link up sensory and motor neurons. So they're basically in the middle, like think intermediate neuron, interneuron. And they associate sensory and motor activity in the central nervous system. And then we have motor neurons. So these have an extensive network of dendrites, large cell bodies, and long axons. And they're much larger and more complicated because they send signals from the brain and the spinal cord to the muscles. All right, there are also important supporting cells that are good to know. These are called glial cells. So here is some info about the 5 different types of glial cells. I'm not going to go over them in too much depth, but I just wanted to point out what the main functions of glial cells are and why they're important. Let me try to get that all in there. So glial cells are the support cells of the nervous system, like I mentioned. They support neurons and they help them carry out the task of transmitting different information around your brain and body. So they don't actually transmit any information themselves, but rather they have a number of really helpful functions, including binding neurons together. They provide structural support for neurons. They also give neurons nutrients and protection. And then they help repair neurons and clean up waste products that are basically the product of neurotransmission. So they're very important, and it would be good to know the different kinds and how they specifically contribute to the support of a neuron. All right, so chapters 4 and 5 essentially cover communication, and I would say right at resting potential right here is where chapters 4 and 5 start. So the difference between these chapters is that chapter 4 mainly covers communication within a neuron, and chapter 5 covers communication between neurons. So I'm going to spend most of my time for this chapter, chapter 4, covering action potentials and how these electrical signals carry information that is sent within a neuron. First, before I start, I wanted to review how the movement of ions create electrical charges. So this information is not on the study guide, but it is on page 116 of your textbook if you did want to open that up at this point to follow along with me. So it's on page 116. So first, the concept of a concentration gradient is really important to understand. This principle states that molecules move from more concentrated areas to less concentrated areas. So for example, the book talks about it, and I think Dr. Levins also talks about it in her lecture. If you dropped a little bit of ink into a cup with water, the ink is going to start off being very concentrated, but it's gradually going to spread out until it's equally distributed. The second important concept to know is a voltage gradient. So this principle states that an ion will move down a voltage gradient from an area of higher charge to an area of lower charge. So sort of the same concept where molecules are moving from a high area to a low area. And these two gradients are important to know because they help us understand how the movement of ions can create an electrical charge that is sent down the axon of a neuron. So this brings us to resting potential, which if you're following along in the book, starts on page 119. So before an action potential, which is that communication within a neuron, before that begins, the neuron is at a resting state called resting potential. And a resting potential is the electrical charge across the cell membrane in the absence of stimulation. So this is stored potential energy. That's why it's called resting potential. And it's produced by a greater negative charge on the extracellular side, so outside the cell, relative to the intracellular side. Specifically, the extracellular side of the membrane at resting potential is 0 millivolts, and the intracellular side is negative 70 millivolts charge. So exactly what we just said. So it's a greater negative charge on the outside greater negative charge on the inside relative to the outside. So also when a neuron is at resting potential, there's more chloride and sodium on the outside of the cell and more potassium on the inside of the cell. So since resting membrane potential is maintained at negative 70 millivolts, there are a couple of key components that help it maintain this charge. So the first one is selective permeability, or having a selectively permeable membrane. And this means that some molecules can get through the membrane through active or passive transport, while others can't get through the membrane. So the membrane of a neuron is relatively impermeable, so largely negatively charged proteins stay inside, and sodium, which is positive, which is Na+, stays outside. This leads to that large negative 70 millivolts charge on the inside because you have a lot of positive ions on the outside. Another key component of resting potential is the concentration gradient. So we just covered this principle, so make sure you know how this applies to resting potential and how the movement of molecules based on a concentration can impact resting potential. And then we have the voltage gradient, which we also just covered. This principle means that ungated potassium channels are allowed to flow freely until an equilibrium is reached. And then finally, we have the sodium-potassium pump. That's another important aspect of resting potential. This comes into play when the sodium-potassium pump gives out sodium, which is positive, from the intracellular fluid, and it also injects potassium. So those are kind of 4 concepts that you'll want to keep in mind for resting potential, which is the state of a neuron before an action potential is created. So now going through the steps of an action potential, so I'm going to kind of come down here. We're going to start with the all or nothing law, which is very straightforward. It's basically the idea that once a neuron is excited to a certain degree, meaning that it hits threshold, which we'll talk about, then it will fire and there is no stopping the neuron at that point. So an action potential is a large, brief reversal in polarity of an axon. This is how a neuron sends information within itself. So it's very short. It lasts about 1 millisecond. And I wanted to go over the steps of an action potential that are briefly listed here, but they are also listed in your textbook, starting at around page 122. And so I'm going to go through them very stepwise, and we'll kind of just put our cursor along here to say where we're at. So like I just mentioned, a neuron is at resting potential before an action potential starts. That means it starts at negative 70 millivolts. So we can kind of see right here that this resting level is just below threshold. So that's at negative 70. At this time, sodium and potassium voltage-gated channels are closed. So we can see these right here. Then depolarization occurs when some sodium ion channels open. So remember, sodium is a plus, it's positive, and depolarization means that the neuron is getting more positive, or if you want, less negative. So sodium channels are coming in, and this is making the intracellular charge more positive, which is increasing this level right here. So when threshold is reached, which is about negative 50 millivolts, all of the sodium ion channels open. And this is when we go into this period. So that means it's getting very positive at this point. So after that, the potassium voltage-gated channels open. And then the membrane hyperpolarizes to ensure a one-way direction for the action potential. And this is called the absolute refractory period. So just like depolarization was becoming more positive, hyperpolarization means that the charge is getting more negative. So the potential, the resting potential, or I guess the charge potential drops slightly lower than resting potential at this point. So as we can see during the relative refractory period, we are dropping lower than this negative 70 millivolts right here. And like I said, this ensures that the action potential is a one-way direction. And then finally, after our hyperpolarization stage, the sodium-potassium pump works to restore the resting concentration gradient. So we go back to that normal negative 70 millivolts and that distribution of sodium and potassium that we had when we were at resting potential. And chloride. Don't forget about chloride. All right, so that kind of took us through the steps of an action potential. So I wanted to also cover briefly postsynaptic potentials. So I wanted to talk about how neurons integrate information and specifically about how excitatory and inhibitory postsynaptic potentials work and what they are. So excitatory postsynaptic potentials, or EPSBs, are a brief depolarization. Remember, this means that the neuron is getting more positive And if it gets more positive, it's more likely to reach threshold, which means that it's more likely that an action potential will happen. So EPSBs are when there's a depolarization of the neuron membrane in response to stimulation, and this makes the neuron more likely to produce an action potential. The exact opposite is an inhibitory postsynaptic potential, which is an IPSB, and this is a brief hyperpolarization, making it less likely that a neuron will produce an action potential. So if we remember what we were talking about with hyperpolarization, that means that the cell is getting more negative, meaning that it's less likely that it would reach that threshold level of negative 50 millivolts, because it's either going to stay at negative 70 or even maybe reduce a little bit. So postsynaptic potentials sum together when they are close together, and this is an important concept to know. So there's two ways of summing postsynaptic potentials. The first one is called temporal summation. This is when two postsynaptic potentials happen close together in time. So for example, two EPSBs could be very close together in terms of their temporal sequence, and this will make them combine into one large EPSB. The other way that postsynaptic potentials can be summed is through spatial summation, and this is when two postsynaptic potentials occur in a similar location. So if the postsynaptic potentials are located close together, then they will be summed to make one large EPSB or IPSB. All right, so that kind of takes us through Chapter 4 information. This chapter that we're moving to, Chapter 5 now, covers communication between neurons. So I'm going to review neurotransmitters and how those get transferred from one neuron to the other. So first I wanted to review the criteria for identifying a neurotransmitter. So I'll just scroll down to put this up at the top. In order to be a neurotransmitter, and this information, if you want to follow along in the book and get a little more detail, it's on page 150. So to be a neurotransmitter, first the chemical must be synthesized in the neuron or be present in that neuron. And then when the neuron is active, the chemical must be released and produce some sort of response in the target. This same response must be obtained when the chemical is experimentally placed on the target. So this could happen through drugs and psychopharmacological processes. And then there must be a mechanism that exists for removing that chemical from its site of action after it's done having that response. All right, now I just wanted to go over also the 4 steps of neurotransmission. So this is not on the study guide, but it starts on page 145. And this is important to know because it kind of ties everything together with communication between neurons. So the 4 steps of neurotransmission include, number 1, synthesis. So during this stage, some neurotransmitters are transported from the cell nucleus to the terminal button. The second step is neurotransmitter release. So in response to an action potential, the transmitter is released across the membrane by exocytosis, meaning that it's exiting the terminal button. The third step is receptor site activation. In this step, neurotransmitters cross the synaptic cleft and they bind to a receptor. And then the final step, the 4th one, is neurotransmitter deactivation. And this is when the neurotransmitter is either taken back to the terminal or it's inactivated in the synaptic cleft.
Okay, so now we're going to get briefly into Chapter 8, which focuses on brain development. So you're going to really need to know the 7 processes of brain development and when they occur. So the first stage is cell birth, when stem cells are formed in the neural tube. It typically starts around the 7th week in utero and is primarily finished at about 5 months. So this is called neurogenesis for when neurons are growing, and the process of forming glial cells is called gliogenesis. So the second stage is cell migration, when at about 7 to 14 weeks in utero, these newly formed neurons migrate to the brain through fibers created by radial glial cells that go to the cortex. And then the cells organize themselves into the brain structures. And then once those cells are in their appropriate place, they differentiate to become specialized based on where they are. So this is pretty much complete by the time of birth. Afterwards, the cells mature, so they can continue for years and they go up into adulthood, but it begins around the 20th week in utero, and it's basically the growth of other parts of the neurons, so the dendrites, axons, and synapses. And then when the axons and dendrites begin to form, so do the synapses, so that's called synaptogenesis, which is when the synapses are formed between the neurons within the nervous system. So throughout life, synaptic pruning also takes place. This is when some neurons and synaptic connections which might not work well are eliminated or pruned to make way for higher quality neuronal connections during times of cognitive development. And then there's also myelinogenesis, which is the formation of myelin around the axon, and it starts prenatally but also continues across the lifetime. And in myelinogenesis, myelin is formed on the axons that are most used, so the ones that are used often, and it increases the speed of impulses in the brain.
All right, so that wraps up our first virtual review session for Exam 1 in Brain and Behavior. I hope this was really useful for you guys, and please, continue to use all of the lectures, your notes from the class, the book, and the study guide that we gave you to review. Remember that you have a 72-hour window that the exam will be open, and once you start the exam, you'll have an hour to complete it. So make sure you're prepared and in a good environment and a good attitude to take the test, and good luck!
Good luck!